Thank you. We welcome you to our one of our SERN Lunch and Learns. We are really thrilled that many of you are joining us from different parts of the country, and there's a lot of interest in this topic. So I'm totally delighted. Many of you are joining us for the first time, and I just want to start off by acknowledging that this series of our Lunch and Learn are sponsored through our HRSA grants from Southeast Regional Genetics Network, where I serve as a PI. And but the topic today is very exciting from newborn screening and from clinical perspective. And we have a very great scientist here from Emory who have admired her work and her rigorousness with the sciences. And I think this kind of work really, we as clinicians and working in the lab strive for, because it helps build evidence to what we do daily, what we bring for our patients. So I'm going to let our project manager, Teresa, introduce Dr. Fridovich-Keil before we start. And Teresa, please help me introduce Dr. Keil.
Hi, good afternoon, everyone. So Dr. Fridovich-Keil is currently serving as a professor in the Department of Human Genetics in the School of Medicine here at Emory. She was born and raised in Durham, North Carolina, where she completed her undergraduate training at Princeton University in biochemistry. And then she moved to Cambridge and completed her PhD in biology at MIT, followed by a postdoc as an ABMG fellowship training at Harvard Medical School. And then in the fall of 1991, Judy moved to Atlanta, where she joined the faculty here at Emory. Her research focused on genetic and environmental modifiers of birth outcomes, including genetic disorders related to galactose metabolism and prenatal environmental exposure. Today, she will be presenting on rethinking Duarte galactosemia.
Thank you so much. So first, I just want to thank you for the kind invitation to speak with you guys. This is a topic that we've cared about for a very long time, and I'm delighted to share with you what we've learned. Okay, so let me see. Okay. How do I get the slides? Yeah, thank you very much, Blake. Okay, so rethinking Duarte galactosemia. So in the next about 30 minutes, here are the topics I'd like to cover. So what is Duarte galactosemia? I'm guessing that most people are probably familiar with it, but for the few people who aren't, I'll explain what it is. What was known about DG before we did our study, and then I'm going to tell you about a 3-year study that we recently completed. And I'll explain what were the goals of this study, what we did and what we found, and what we think it means and why we think it's important. Okay, so before I go further, I just need to say that this project was a huge team effort. As you'll see, there were a lot of children and families. Corey was our funding agency. We had a stakeholder executive committee that involved people from support groups, parents, healthcare providers, newborn screening people, and so this could not have happened without all, excuse me, I'm sorry, turn that off. Hopefully that will not happen again. Okay, so sorry, this was a huge team effort. Okay, so what is Duarte galactosemia? So DG results from partial deficiency of an enzyme that we're going to call GALT, G-A-L-T. That's actually galactose 1-phosphate uridyl transferase. So it's the red one. In this pathway of galactose metabolism, this is the lumbar pathway. And most people who hear about this enzyme actually know that it is also deficient. It is profoundly deficient in patients with classic galactosemia. And so this is a slide which is just meant to help people get oriented to what are we talking about in terms of Duarte versus classic galactosemia. And so here, these are box and whisker plots showing red blood cell GALT activity. So these are actually values from a clinical lab. And you can see the GALT genotypes on the bottom, and the red arrow points to samples from patients with classic galactosemia, and they have pretty much undetectable or almost undetectable GALT activity, whereas the blue arrow points to samples from patients with Duarte galactosemia, and they have on average about 25% of what we call the normal level, but recognize that the normal level is actually quite a range. It's not, a very discrete number that's the same in everyone. Okay, I'm not going to spend a lot of time in this, but what I wanted to point out is that, so we've actually done a lot of work looking at the Duarte allele, which is found in everyone with Duarte galactosemia. So first, let's just look at the little pedigree over there. And patients who have Duarte galactosemia actually have two different alleles of GALT. One of them has a mutation which profoundly impairs function, and we call that the G allele. The other one is what we call the D2 allele, and it actually has a number of variants. One of them is a 4-base pair deletion in the 5' region of the gene that impairs the expression, and then there are some intronic changes, and there's also a silent mutation. And we looked, so N314D is an amino acid substitution, which is found in Duarte galactosemia, in the Duarte allele. And people used to think that that was a mutation. And so we actually looked, and what we found was that so N314D, D is actually the ancestral allele. And if you look at non-human primates and you look at other mammals and you look in other species, basically what you see is N is the mutation, D is not the mutation. And we could even trace to see when that arose. So it arose early in human development. And basically we looked at the different variants, and we could see by looking at comparative genomics when the different variants arose. And the reason why I wanted to show this slide is that you'll see that the D2 allele is actually very common in people of European ancestry, and it's much more rare in people whose ancestry is not from Europe. So it looks like it probably arose, this part of it, the 4-base pair deletion in the 5' region of the gene, likely arose in a population of humans who had already migrated to Europe. And so when we look at newborn screening and look in populations where we have mixed ancestry, if you look at the ancestry of the infants who are identified with Duarte galactosemia, they're like 90+% of European ancestry, and this is why. Okay, so here, this is a slide showing results from newborn screening over multiple years. And so the little numbers, which I don't expect you to be able to read from the back of the room, but there are numbers over each of these bars, and those are actually the years of data. So the number of years, so there were 16 years of data that went into making this particular part of the graph, 15 years of data here. So these are the states down here who responded to our inquiries. And what you can see is that of states that are identifying infants with classic galactosemia, although the numbers bounce around, it's about 2 per 100,000, which is about 1 in 50,000, which is actually what we kind of thought it should be. For Duarte galactosemia, it bounces around even more. So in New York, they don't have it. It's not because it doesn't exist, it's because they don't detect it. So how much Duarte galactosemia is identified is really influenced by how the newborn screening is done and where they set their cutoffs. Okay, so this was one of the observations, that Duarte galactosemia is identified in some states, not identified in others, and certainly not completely identified probably anywhere. Now the third panel shows you false positives. So those are infants who had a positive initial newborn screen for galactosemia, but who on follow-up testing were found not to have galactosemia. And you can see that in some states, there are really high false positive rates. And that's gonna come back. Yes.
Good question. So is the reason that New York is undetected, I mean, they don't find cases, were they doing total galactose instead of the enzyme activity? And that's why
I believe they're also doing the enzyme activity, but they set their cutoff
so high.
intentionally so that they would not pick up Duarte.
Okay. Thank you.
Okay, so why are we talking about Duarte galactosemia? Well, it exists. It's been identified by newborn screening for a very long period of time, and there have been differences of opinion about how should these babies be treated. So we know that infants with Duarte galactosemia generally thrive while drinking milk. That's either breast milk or milk-based formula. There was no strong direct evidence of long-term complications. There were some indirect evidence. And we know that breastfeeding has a lot of known benefits. And so the question is, do we want to interfere with that? Sometimes, obviously, we need to, like in classic galactosemia. Should we be interfering with breastfeeding for Duarte galactosemia? The reasons for thinking maybe we should, we know that newborns with DG accumulate elevated levels of galactose metabolites when they're drinking milk. And we also know that children with classic galactosemia, even if they don't drink milk as infants and don't have acute symptoms as infants, can still go on to experience long-term developmental problems. And so the concern was, well, just because this kid with Duarte galactosemia seems to be thriving now as an infant, drinking milk, we know their metabolites are high, not as high as if they had classic galactosemia, but it's the same set of metabolites and they are abnormal. Are we putting this child at risk for longer-term developmental complications? So that was sort of the unanswered question. So what was known about long-term outcomes in DG before our study? So these really were the three main studies that had been published. And without going into a lot of detail, one of them basically looked at a fairly small number of subjects who were really very young. A lot of those kids were actually 1 year old. And they did some developmental assessment and they said, we don't see anything wrong, but they also had no control group and they did no adjustment for covariates like socioeconomic status. And then the Powell study that was from right here in Atlanta, some of the authors are in the audience, had the benefit of being a population-based study, but it was also an indirect study, meaning that they weren't directly assessing developmental outcomes. They were asking how many children received intervention in school for given outcomes, and are kids with Duarte galactosemia overrepresented or not? And so there it looked like, well, maybe they are overrepresented. Maybe there is a problem, and that really is what inspired us to say, okay, we need to figure this out. And then that was a study actually from our group that was looking at a hormone marker of ovarian function in girls with Duarte galactosemia, and we saw absolutely no difference between the DGs and the controls. Okay, so healthcare providers and parents were faced with these decisions. The baby is identified by newborn screening. They are determined by follow-up testing to have Duarte galactosemia. What should you feed them? Should they drink milk or should they drink soy? And so we said, hey, there's this naturally occurring experiment going on where kids are being identified and some of them are being told to drink milk and some of them are being told to drink soy. And then they're growing up, and all we really need to do to answer this question is to find them and actually do developmental assessments on them when they're old enough that if they're going to have problems, hopefully we would see it, and then compare. So we had two aims to our study. The first one was to test a whole bunch of kids with Duarte galactosemia, and we focused on children who were between 6 and 12 years old. And we wanted to know, do the kids with DG demonstrate an increased prevalence of developmental problems? And then the second aim was so here we're comparing cases versus controls. Here, we're only looking at the cases. We're only looking at kids with Duarte galactosemia, but we're looking at kids with Duarte galactosemia who drank milk as babies versus who drank low galactose formula as babies, and asking, are their developmental outcomes any different? Okay, so it's very, very simple design. Okay, so this was an observational case control study, and this is just a diagram that I made to sort of help illustrate. So we recruited a bunch of families on the basis of them having at least one child who had been identified as having Duarte galactosemia in one of the states that was participating. We had 18 different states. 17 states plus the District of Columbia that we were recruiting from. And we recruited families with the assistance of either the newborn screening follow-up people who sent out our recruiting envelopes. And in some states, the newborn screening follow-up person couldn't help us, but they turfed us to the metabolic clinics that had done the actual diagnosis, and they helped us. So I'll explain a little bit more about how we actually did the recruiting, but we found families whose children were now 6 to 12 years old who had been identified as infants as having Duarte galactosemia. And Duarte galactosemia is an autosomal recessive condition, so those same families also, if they have other children, are likely to have some children who are not affected. Okay, and of the kids with Duarte galactosemia, so those are the shaded symbols, some of them drank milk and some of them drank soy. And so we were going to recruit a whole bunch of them and test the ones who were in the right age range, and of course the testers would be blinded, so they wouldn't know, they would know this is little Joey, but they wouldn't know whether Joey has Duarte galactosemia or not, and if Joey has Duarte galactosemia, whether or not Joey drank milk, right? So, so they would basically just test a whole bunch of kids, and then we would take the results of those tests and we would compare the cases versus the controls, and then in the second comparison, we would test the cases who had drunk milk to the cases who had drunk soy. Okay, so simple design. Okay, so again, we did initial recruitment, obviously informed consent. Then we had part one, which is we gathered a lot of information using online or sometimes telephone parent-guardian surveys. And there we were gathering information about the family, the demographics, a bunch of covariates that we thought we might need to control for, and importantly, diet information about the child actually at different points when they were an infant, when they were a young child, and more recent. And then from the people who participated in part one, we also recruited for what we'll call part two, and that was the direct assessment part. So we actually sent a team of child psychologists, speech pathologists, and Grace, our project manager, who coordinated that part, out. We rented space in a bunch of different locations in all these different states, and they set up schedules and did a lot of testing, and then we had a ton of data, and we did data analysis. Okay, so here's where we recruited from, and so you can see, I think the map is very useful in helping you sort of visually see that we had, from the Southeast, these were our two sort of big ones, Texas and California. Anyway, so we recruited basically every state that would talk to me. we did our best to bring into the study. Okay, so how did the initial recruitment work? So in my lab, we just took a bench and we basically were folding and stuffing envelopes. Unfortunately, we had a bunch of undergrads, and so we folded and stuffed a lot of envelopes. So we would have a particular state. So for instance, in Alabama, we asked, okay, these are the target years. This is when the kids would have been born for them to be in the age range that we're interested in now. So please look back at your records and tell us how many names and addresses do you have for infants who were identified with Duarte galactosemia in those target years. And so don't give us the names or the addresses, just tell us the number. So for Alabama, they said, well, we have 105. So we prepared 105 envelopes that were stuffed. So they had a cover letter from the group that was actually going to mail the envelope, basically saying, we have not violated your privacy. We didn't give out your name to anybody. Had a cover letter from us, and then it had kind of a simple bullet point, what the study is, and if you're interested, how to reach us. Okay, so we would prepare a bunch of envelopes, and what is important is that the return address was actually to our colleague at the recruiting location who was going to mail the envelopes, because as you might imagine, a lot of these addresses were from when the person was an infant, and now the person was 6 to 12 years old, and sometimes families had moved. So a lot of these envelopes were going to bounce back, and we didn't want them to bounce back to us because then we would see the names and addresses on them. So we prepared a bunch of envelopes that had everything except the address of who it was going to, and we put them in a package and ship it to the person who was going to print out the address labels, stick them on the envelopes, and put them in the US mail. Okay, and those people who received the envelopes, if they were interested and wanted to learn more, we had a phone number and an email address, and Grace was the kind person who they would generally reach when they called that phone number or sent that email, and then she would tell them more about the study, and we would go from there. Okay, who participated? So we received completed Part 1 surveys for 567 children, and 350 children completed the full study. So that's Parts 1 and 2. So you might ask, how come the 567 is a lot bigger than 350? So part of it is that there were some geographies where we had 4 people who responded who were within, a 2-hour drive. Our budget was not big enough for us to actually send our team to that location to test 4 people. So part of it was geography. Did they live close enough? Part of it was sometimes we learned that from their Part 1 survey, they had some confounding issue. We had one child with shaken baby syndrome. We had, these were very, very small numbers, but we had some children who had other issues that we thought really would not, would confound interpretation. And so most people were excluded because of geography. And sometimes, our team was going to be there on these 4 days, and they were going to be visiting grandma, or, so there were all kinds of reasons, most of which had to do with geography and logistics. Okay, so this tells you a little bit about the 350 who participated. So 206 of them were cases, 144 of them were controls. And again, the controls were unaffected children from the same families, but we were not doing a pairwise comparison. So we were not saying, okay, we've got these 2 kids from the same family and we're going to compare their outcomes. We're saying we've got a bunch of families. We're going to take all the cases from those families and they go in this group, and we're going to take all the controls from those families and they go in this group, and we're comparing the groups. Okay, so that's important. Of course, when we did the statistics, we used appropriate statistical tools that took into account the fact that there is, shared alleles between cases, between siblings and so on. Okay, so you can see that our cases and controls, our average age was a little more than 9 years old. For dairy exposure, about 40% of our cases had been exposed to dairy, and that was when they were less than 1 year old. 91%, most of our controls had drunk dairy. And then you can see that we had a pretty reasonable distribution of boys and girls in this. Okay, what information did we gather? Okay, so from those first surveys, we gathered demographic information, some socioeconomic status information about the family, other covariates, and information importantly about the child's diet. And then by the direct assessments, these are the areas of development that we tested. So thinking and problem-solving skills, socio-emotional development, physical measures like height, weight, head circumference, auditory processing, speech and language development, and physical development, which was movement and coordination. And all of these things were tested using established, so we didn't just make up our own tests. There are plenty of established assessments that we use that are normed and so on. This is what a direct testing block looked like. So from the perspective of the child, when the child came in, they were given one of these cards, and they would go to the different stations, and in one station they might see a speech pathologist. And a lot of what we did, we were doing actual assessments, but we tried to make it in sort of a game-like context so that the kids would enjoy it. So they would go to station 1, and the person who was at station 1 had stickers, and they would put a sticker in that space, and they would initial it, and that way we knew that that child had seen, you know, which tester they had seen and in what order. So anyway, so they went to the different stations, and when they finished, they got to go to the toy chest and get a really cool toy. So that's what it looked like for the child. This is what it looked like for the testing team. Okay, so this was the daily schedule. So the team could test up to 6 children in a day, and basically they were sort of staggered, and we had breaks in there and where the kids could have a snack, and of course a parent came with the child. We had some additional surveys that the parents filled out while the kids were getting tested, and so anyway, so it was busy, but it worked well. And then at the end, we also had feedback forms that we would ask the family to fill out, like, you know, how did it go, and did it, you know, what worked well, what could we improve, and so on. Okay, these are the actual assessments. I don't expect you to read this, but the bottom line is we had assessments for each of the different developmental areas, and they are sort of the standard ones that have been normed and that are used clinically. Okay, so what did our data look like? So again, we had 350 subjects who completed the full study, 206 cases, 144 controls. We confirmed the case control status by Gault genotyping, and we actually sequenced the entire about 6 kb of DNA for every child who participated in the direct testing part of it. And we did that from saliva samples. And that turned out to be extremely important because about 10% of the kids, you know, when we started out and we thought, you know, this is little Joey. Is little Joey a case or a control? We had asked the parent, was this child identified, you know, by newborn screening as having Duarte galactosemia? About 10% of them were not what the parent thought they were. Most of the time, we had a child who the parent said, they never told me my child, you know, the newborn screening did not come back positive. We did not have follow-up testing. It wasn't until my younger child was born that, you know, that child was identified with Duarte galactosemia. They thought their older one was a control, and it turned out their older one was also a case. And we had a few where it was the reverse, where they had been told, we were told this child had Duarte galactosemia, and when we sequenced the DNA, they were actually a carrier or they were a DD. So whenever we had a child who was not what the parent had told us they were, of course, we wanted to, you know, we did follow up, and I called them, and in general, the children who had been missed either were born, remember, these were kids who had been born like up to 12 years ago. So some of those kids were born at a time when the lab was only doing a GALT activity. They had no other supporting data to make a determination on, and they were making the best guess they could with the information they had. But if you remember from that first graph I showed you with those box and whisker plots, there's actually overlap between what the GALT activity is in some of the Duartes and in some carriers, for instance. And so you know, these were very honest mistakes. So some kids had been missed by newborn screening, and some kids had been, you know, called as A when in fact they were B. Okay, so everybody, all 350 who we included in the final analysis, you know, had been confirmed as either a case or a control. We had outcome measures. We had numbers. We had 73 outcome variables that had been quantified per child, and these were in the 5 developmental domains. So we, you know, did a lot of testing, and you end up with a lot of variables. And so we had 73, and we also had a whole lot of covariates that we had collected, and so we could ask, are these, you know, do we need to adjust for any of these covariates? Okay, so how did we analyze the data? So first, all the statistical analyses were performed using R. First, we did descriptive statistics to compare demographics between groups and to identify covariates that basically were different between groups that we were trying to compare, so we needed to adjust for those. We also tested for what I'll call missingness. Now, we didn't have a lot of missing data, but occasionally we had a child who, for one particular assessment, one of the variables had not worked well, and it was missing. And so we wanted to make sure that was that missing, that like were more cases, did we see more missing variables, you know, for variable A among cases than controls? And so we tested that, and in fact, the missingness was kind of random between cases and controls. And then we ended up splitting our data set, and we split it, about 75% of the kids went into a group that we called the validation set, and about 25% of them went into a group that we called the discovery set. And so you can see it was actually 263 kids were in the validation, and 87 were in the discovery set. And we did that because when we originally designed this study, we said, okay, bottom line is we had not sort of appreciated how many independent variables we were going to have per child. And we did not have, our study was not powered to test 73 outcomes on every single child. So we had a whole bunch of meetings, and of course, the speech pathologists thought that their outcomes were the most important. And the child psychologists thought that their outcomes were the most important, and so we kept going around and around in circles, and finally decided what we really need to do is just divide up the set, and very carefully divided it up so that it was random and the sets kind of matched each other in terms of the proportion of cases versus controls and the ages. You didn't want a bunch of 6-year-olds in one group and a bunch of 12-year-olds in the other, so we made sure that they were very balanced on all parameters. And then we tested all 73 outcomes for association with DG status in the discovery set. So we're not worrying about multiple test correction. We are just defining p-values and ranking them. Okay, so smallest to largest. And then we took the 10 with the smallest p-values, and those are what we tested against the validation set. Okay, and so we tested them against the validation set for association with DG status. So that was for AIM-1. And then just looking at the cases, at the milk drinkers versus the soy drinkers, we looked at those same 10. Okay, so what did we find? So first question was, did the kids with DG show any more problems in any of these outcome areas than their unaffected than the controls? And the answer was no. And then for AIM-2, did the milk drinking DGs show any worse outcomes than the soy drinking DGs? And the answer was no. And so the statistics were very clear cut, but what I want to show you are just some graphs because I'm a visual person. Okay, so these are just some examples. And so this is an IQ test. Okay, and you can see that our controls, so 100 is supposed to be the population average. A lot of people before us have shown that families who choose to participate in research studies tend to be higher educated and have high achieving kids. So we were delighted to see how smart all those kids were. But I think what you can see is that the kids with DG were actually just as smart or even smarter. And there's a range in both of them, but you can see that the DGs are certainly not any worse off. And here, these are just cases, and they're divided up into the soy group and the milk group. And again, you can see that if anything, the milk drinkers are doing better than the soy drinkers. This wasn't statistically significant, but milk certainly did not have long-term IQ problems for these kids. And then this is actually from one of the movement assessments of like coordination and movement. And again, you can see that for the cases and the controls, that they really are indistinguishable. And for the milk drinkers and the soy drinkers, they really are indistinguishable. And we pretty much have 73 graphs that look like this. So yes, we did very careful statistics, but the truth was, really didn't almost need to do this. Just looking at it, it was very clear that there was not a difference. Okay, so I want to recognize our limitations. So first, this was one study. Yeah, it was a big study. It took us 3 years. God, we worked hard. But it was one study. And 350 is a big number for a study of a rare condition, but it's not a huge number. It's not like 10,000 people or something. This is hundreds. The age range, everybody who participated and completed the study was between 6 and 12 years old at the time of testing. So we didn't test younger or older volunteers. So we really can't, speak to outcomes from before or after. Now I'm gonna I'm going to modify that a little bit. We did in some of the parent questionnaires, we did ask the parents, like, did your child have any developmental problems or need any special interventions when they were younger? And so, the answer was it was exactly the same between the cases and the controls, but our direct testing was all just 6 to 12 year olds. The diet information we had, it came from the families, but it was retrospective. So it is always possible that, that the parents didn't remember everything accurately, but, we did the best we could. All of our volunteers were from the lower 48, we're from the continental US. They were almost all white. And I wanna point that out because when we're doing the first states and we're like, oh my God, we're just doing a study of white people. What, are like, what's going on? And then I remembered that study that we had done years ago that the D2 allele is pretty much only found in people of European ancestry. And so I quick contacted the states that had been working with us, and I said, do me a favor, look, do you keep track of race and ethnicity of the babies who get screened? And I was like, yeah, we do. I mean, I'm sure there's some errors in it, but yeah, we do. So well, do me a favor, look back at however many years of data you have easy access to, and that was different for different states. Please tell me, what was the racial makeup of your screened population, and what was the racial makeup of your DG, identified positive population? And in every state that responded to us, they were like, oh my God, DGs are a bunch of white people, and the screened population was quite diverse. So I don't think that we had huge ascertainment bias in who responded to our envelopes. I think that the DG population is largely of European ancestry, and so that's who participated. So if you wanted to do a study of DG somewhere else, I think you would still be looking at a bunch of people of European ancestry just because that's where the D2 was. And finally, we tested 73 outcomes. So, we thought that was a lot, but surely there are outcomes we did not test, and so it is very possible that there were differences in some of those other outcomes that we did not test. Okay, so what do our results mean? So just sort of at face value, our results suggest that children with DG are not at increased risk for detectable developmental problems, regardless of milk exposure as babies. I mean, that's kind of the simplest interpretation of the data that we found. If that is true, then it suggests that DG families should be able to breastfeed with confidence if they choose to do that. Clinicians should be able to give evidence-based prognostic and diet recommendations to DG families. And public health, I think this raises the question of, if these kids really are not at increased risk and don't need intervention, should we be trying to pick them up by newborn screening? Are we doing something good, or are we doing something bad? If we set our newborn screening to try to pick up every possible DG, then those kids are going to come in. They're not going to be told you have DG. They're going to be told you had a positive galactosemia. So many of them are going to stop breastfeeding. If they're breastfeeding, hopefully they'll start pumping, but especially if it's a first-time mother, speaking as someone who has breastfed 2 children, I can say that, if when my baby was 1 week old, you told me I had to stop and just pump, and then 2 weeks later I could go back, I don't know if I would have been able to do that. So the other issue is that when you set your cutoff to try to pick up every kid with Duarte galactosemia, your false positive rate goes way up. That's just sort of the nature of the beast. And so you are pretty much freaking out a whole lot of families. And is that a good thing? So I think that's a discussion really that needs to happen, among people who have expertise in that area. Okay, for anyone who wants to read more about Duarte galactosemia, so our paper, Grace was the first author, was published in January of this year in the Journal of Pediatrics, and we made a 5-minute video abstract. So if you want to hear basically this talk condensed down into slightly less than 5 minutes, you can hear me and Grace talking real fast. You can go to the pediatric site. It was published open access so that everyone could see it. Unbeknownst to me, the journal actually asked Sean McCandless, who at the time was the president of the Society for Inherited Metabolic Disorders, to write a commentary, and so it's in the same journal. And so he did that, and that really helps to put it in context from the clinician's point of view. We have a gene reviews article that we updated, so you can read a lot about it there. We made a family-friendly website that has lots of information about Duarte galactosemia, and because Corey made us, we submitted everything to clinicaltrials.gov, and the results are all there. So if you want to see every detail of how this study was done and what the results were and how they were analyzed, it is all freely available online. And special thanks, so that was our research team. Obviously, we had many wonderful volunteers, hundreds of them, families who participated in this study. We also obviously had many people in the States that were helping us with the recruiting. Brad Thorell and Paul Fernhoff are the ones who really encouraged me to start this. And I have to say, Paul was, for decades, saying, Judy, I see all these patients with Duarte galactosemia, and I really don't know what the right answer is. Somebody needs to study this. Somebody needs to study this. So finally we did, and our funding source. So any questions?